All right, so thank you very much for the opportunity to come back here. It's been 20 years since I've been back. Right when I moved back to the US, Greg Howe had invited me to give a seminar, and I haven't been back in East Lansing since then. My in-laws had celebrated their 60th wedding anniversary, so I had my nice stock slide ready for this celebration to go through. And so, in terms of talking about what big impact the PRL had, I'm going to start with the really obvious one. And there we go. So Antje and I got married here. We met I was a graduate student in Hans Kende's lab, Antje in Natasha Reichel's lab. And just kind of thinking about with the jokes, when we got married, when we got our wedding invitations, I kind of picked up Hans's sense of humor. And so I had taken our wedding invitations and printed on nice paper ones that said that Hans Kende and Natasha Reichel would like to invite you to the wedding of their graduate students. Hans found this amusing as all get out. Natasha freaked out. So it tells us a little bit about the difference in sense of humor of various people that were there. But certainly, this has impacted everything in my life going forward. And so this is a really special place for us and coming back and seeing all the great friends. And since I'm supposed to represent the 90s, this also gives an opportunity to show you some really good 90s hair. Now, in fairness, we were at my friend's wedding up in Minneapolis, and so it was really humid, so that added to it. But, yeah, I have to own that, and you can't unsee that. So thinking about the training we have, and this is something that Antje and I always really talk about, is just the fabulous science training that we had here at the PRL. If you look through that list of the professors giving us the graduate courses and the level of expectation that we were held to. I still remember, I didn't know Doug had been in Jan's lab, but he was on my committee, and so when I went for my qualifying exam, you ask what you're supposed to study. So Jan's response was, read the last 10 years of annual reviews of plant physiology and plant biochemistry, and he was dead serious. So that training was fantastic. And, Hans's line was that, what did it mean to have a Ph.D.? And it really meant that you were now trained to go out and do independent research. And I feel like that's really where I got to from all this great background training that we had. The fundamentals, in a really broad sense, were really well established. But it wasn't really just the professors. It was just an incredible time here in terms of the science. There were so many fantastic people. And I thought about starting to list them, and I was like, I'm going to forget somebody and feel real bad about it. But it was just the level of science going on, what Mary Lou had referred to with the sort of the advent of Arabidopsis genetics, molecular biology. There were just so many fabulous people here, and we were all talking with each other, but it wasn't competitive, right? We were building up each other, and it was, sports analogies fit in everywhere. Nobody improves by being the best player on the worst team. So being a good player at the best team was really important in terms of really rising to the level that one needs to continue on. And I'm really happy that Doug is here. I mentioned to him before, because he had a major impact on my career. So certainly, as we know, Arabidopsis genetics going through really dominated things, and for good reason, right? It really broke down all these barriers of our understanding. But for those of us that were looking at things, I worked on ethylene biosynthesis, ethylene signaling, and what I could see is that there was sort of this gap in terms of getting into the biochemistry, thinking about signaling going on. And we used to have these seminars at the PRL, lunchtime seminars, and one of those was by Doug, who at the time was running the mass spec facility in the biochem. And the importance of this is that mass spec for a long time had been utilized for small molecule analysis, but that transition in terms of the technology was right when the advent of those machines could now start analyzing proteins and peptides. And so he gave this seminar, and so I always point this out, and you'll see why going through. I always talk to younger scientists, when your students say, why am I going to that seminar? I don't work on that. Well, you never know where the seeds are going to get planted in terms of your own studies. So, the importance of this was thinking about that we could now do things, or at least we thought we could do things with mass spectrometry that we could never do before. And being at the PRL, one saw the building of the EST libraries, right? Starting with the genome sequencing with Arabidopsis. And so I was aware of how that was growing. And really, to run any type of proteomics and mass spectrometry, you needed that genomic database. And so starting to put these two together, I really started contemplating whether or not we could start making use of all these new developing tools and technologies to go in and use proteomics to study protein phosphorylation. And so the reason that I have Ken's face up here, he was director at the time. So I remember being at one of our pizza lunches, right, that we had for everybody to come together and talk. And I'm working on fellowships and just writing about this and talking about what we were going to do with proteomics. And Ken, with his kind of wry grin, just sort of looked at me and said, sure you will, right? But if you're going to go off and do something, you have to be confident in yourself. So never took that wrong. And it was always fun because we were joking. We always remember with Ken being in the PRL seminars, and when Ken raises his hand at the end and says, now I may have missed it. And we go, oh, the poor speaker is about to be doomed. So again, it was great to be around the sharp people. So was starting to really just simmer all these ideas about what we could possibly end up doing. And then you talk about the PRL and all the people that were here and people that had passed through the PRL. So I don't remember where this exactly is, but this is, Hans is there. Mike Sussman was a graduate student in Hans's lab, so he was back for some reason. I realized I had to label that because as I was looking at it, perfectly fine, and then I realized, that's not inherently obvious to everyone. That was me 30 years ago. So I figured, okay, we're going to slap a name on that. But hiding in the background, that little head in the background, was Thomas Böhler. So Thomas had been a postdoc with Hans, and then he's the director at the Botanisch Institute in Basel, as well as at the Friedrich Miescher Institute. And as I was contemplating about getting into proteomics and starting to think about this field that wasn't even named yet, phosphoproteomics, going through the literature, Thomas had the only evidence I could find to see rapid pulse labeling with radioactivity. So the idea being that if we wanted to look for proteins that were differentially phosphorylated during a response, I needed some readout to be able to trace it. And so looking at that, that was in the early 90s. They did this with radioactive pulse labeling with plants, ran it out on 1D gels, and so that was kind of it. It was descriptive. But so I took that idea and sort of ran with it. And so wrote a postdoctoral fellowship to go to Thomas's lab at the Friedrich Miescher Institute. And that was right at the same time that Thomas's lab was first discovering flagellin and the flagellin receptor. So that was with no background in it, but purely because it was a biochemical tool, suddenly I was working on plant innate immunity. So initially, this was the 2D gels that I was using to at least do proof of concept. We could do 30-second pulse labeling. I was going through 10 millicuries of P33 a week. And what you can see here, for instance, on these high-resolution 2D gels, the green shows you that there's constitutively phosphorylated, constitutive turnover of that phosphate. In the blue, you see, it's the red that can become rapidly phosphorylated. You see the increase in radioactive incorporation. The blue shows you things that come a little bit later biochemically, meaning more than 2 minutes. And so the trick was not getting the radioactive labeling. The trick was building the tools to start getting enough of those to sequence. And those was back in the day before we had LCMS. So we're doing direct infusion into the LC, collecting the data and going off on their computer searches as we're going after it. But this really yielded the proof of concept that we could really go and do what we thought. We got a number of protein candidates that we then could confirm differentially phosphorylated, making use of the peptide sequence tags because of the genome information here at the PRL. But this is really at the advent of proteomics getting utilized in plants, and so having that in pocket got me a recruitment to the Sainsbury lab in Norwich, where I spent 5 years. And the idea there was really to get past the limitation of 2D gels. So the 2D gels worked, but 2D gels just have all the biochemical limitations that they always had. They don't work well with very acidic proteins, they don't work well at all with basic proteins, and they're not great with hydrophobic proteins. And so, we had already started coming up with some ideas during my postdoc, and when we started the lab, what we came up with is really what turned into the basis of all phosphoproteomics. So we're isolating plasma membranes, and part of our purification scheme doing aqueous two-phase partitioning was using Bridge 58, a polyvinyl ether detergent. And what we knew, we were using that as a cleanup step, and what that does, and it's detergents, right? It's black magic, we don't know how they work, but it completely inverts plasma membrane vesicles. And why that was important is, that means we could isolate plasma membranes and then turn them inside out, and then we could shave with trypsin and get all the peptides off of the plasma membrane vesicles, leaving all the hydrophobic domains behind, leaving all the lipids behind, ultracentrifuge out those vesicles so we have the peptides, and then we were going to use IMAC. And so IMAC, immobilized metal affinity chromatography, is inherently sort of a simple idea that the strong positive charge of the transition metal would bind with the negative charge of the phosphate and pull it out of solution. Now that had been used for individual proteins, but what was sort of the hang-up was in trying to use this in bulk was that proteins have all those acidic residues, the aspartate and glutamate. Well, we came into this with what turned out to be a relatively simple concept, which is the pKa's of phosphate in those acidic residues are very different from each other. So we reasoned that if we could get the buffer sufficiently acidic, we should be able to protonate the amino acids but leave the charge on the peptides. And that's what we did. We also had to come up with a column chromatography that acidic condition wouldn't disintegrate. But eventually we could really work this out such that we could get 90% pure phosphopeptides that we could then sequence and find out not only what proteins were phosphorylated, but exactly what residues were phosphorylated. So my lab in the UK, this is the first time anybody did this in any organisms. We beat the mammalian labs, the yeast labs, by working out the chemistry on this. And in that one experiment, we got 10 times more phosphorylation data from plants than the rest of the history of plant biology. And that's just the power of mass spectrometry. So then we wanted to be able to utilize it because, of course, the whole purpose of this wasn't in and of itself just collecting a database, but get functional information. And to do that, we needed quantitation. So, for instance, here's the NADPH oxidase that produces the reactive oxygen when the plant undergoes an infection. So we knew the enzymes were there, but the analogy to the mammalian broke down because the regulatory proteins to activate this enzyme weren't present in the plant genome. So we knew it had to be something else. So we found phosphorylation sites out in this unique N-terminal portion on the protein. And the way that we did quantitation without getting into it is we use isobaric tags. And so the nice part about that is you can label your samples from time points, for instance, on a time course, and then pool everything together. When you fragmented to identify your protein, you got out then this isobaric tag. And what we could see, for instance, on this peptide with the phosphorylation, that sequence, that quantitation doesn't change much, but this peptide actually massively increases in phosphorylation. And so we could go in then and do complementation of the mutant and show that phosphorylation of these residues is necessary but not sufficient to activate the enzyme. And those turned out then to be the residues that Cyril Zipfel's lab later found to be phosphorylated by BIC1. But this now gave us the entire tool set to really go in and start dissecting and finding out not only which proteins are being phosphorylated, but exactly on which residues. So the analogy I like to make is it's sort of like opening a big puzzle box. You had all these new proteins, and particularly at that time, a shocking number of them were unknowns, and you didn't know what they ended up doing. And the problem in trying to put these together into some sort of coherent message is that we didn't know what resistance was. So if you have all these puzzle pieces, right, you start and you make the border. Well, we had no border. And the real kind of connection and breakthrough on this came from sort of this simple model. So, as I alluded to in the Bowler lab, they had just discovered the flagellin receptor. And so the idea is, just like in animals, that plants have these cell surface receptors that are monitoring the environment, and they're detecting foreign invaders, these non-self organisms, through peptides, proteins, small molecules that would be present in the microbe, not in the plant. And that non-self recognition would trigger then all these downstream responses and do that enigmatic stopping of the pathogen. And that's the part that we really didn't have any idea what it was at the time. But the real clue to this, and what's really important then, is thinking about the fact that if you look at bacterial growth, colony-forming units after the infection, whatever this mechanism is, it has to be bacteriostatic rather than bactericidal, right? Because it's not killing the bacteria. The bacteria are simply stopping to grow. So if we're looking for that mechanism, whatever we find has to be consistent with that idea. And the key to this turns out to be then, as our thought process evolved, thinking about the fact that, yes, the plants are undergoing non-self-recognition, but the bacteria are doing the same thing. Microbes spend the majority of their life in the soil competing with other microbes. And only when they recognize they're around a potential host do they activate their type 3 secretion system. And so that needle-like pillar structure injects 20 to 30 effector proteins that shut down the immune response, gets rid of whatever that resistance is, and allows the bacteria to grow and propagate and infect the rest of the cells and eventually end up with disease. So coming back to this, about which pathways were utilized, we employed some technology that really sped up things because reverse genetics, when you're trying to do pathology, is slow. But what we could do is we transformed the Pseudomonas syringae pathovar tomato DC3000, a strain of the Pseudomonas that infects Arabidopsis, transformed with an operon from autoluminescent bacteria. So what we could do is spray it onto our plants and screen through larger numbers, because we could put that directly under photon imaging cameras and monitor the bacterial growth in real time. So what we see here was the real key mutant in getting into all of this. Here we see on Vacisleskia wild type bacterial growth. We see the light there. On this MKP1 mutant, a MAP kinase phosphatase 1 mutant, we don't see much of that bacterial growth, and we could show it's coming from that gene through complementation. We get the growth back. And why this was such an interesting and exciting mutant is what existed in the rest of the defense field. We had a number of mutants that could have enhanced resistance against bacteria, but all of them went through either pre-activation or hyperaccumulation of salicylic acid. The problem with that was, yes, they were resistant, but they have that fitness penalty from the pre-activation of immunity. And you're never going to go to a farmer and say, I can guarantee you resistance, but I'm going to guarantee you 20% yield, right? So that wasn't going to be a viable solution. And we don't see that fitness penalty here. And in fact, we went on and we could show by doing double mutants with all the SA pathway that this resistance is completely independent of salicylic acid. It's doing something else. And what is that something else? It comes back to this idea of non-self-recognition. The bacteria need to recognize the potential host to activate its virulence program. And what we did was worked out assays to be able to look at that signaling, and what we could find is the MKP1 mutant does not produce the chemical signals the bacteria need to actually transition into a pathogen. So what we had essentially discovered are stealth plants. The bacteria land, but they never see it, and therefore they never become pathogenic. But this now gave us the genetics to really start dissecting that chemical communication. So we could go in and do GC mass spec, and this is not grinding up the plant. The bacteria land on the surface, going through the stomates. So these chemicals, we reasoned, had to be present on the surface of the leaves. And so we could take the exudates from the leaves, and this is just a small portion of the GCMS trace. And so thankfully, not everything changes, right? It's a nightmare scenario when your mutant is completely different in every way from the wild type, but plenty of things were there. But in the MKP1 mutant, there are lots of chemicals that were either decreased or even absent compared to the wild type. And we could do this to narrow down candidates. We had 70 chemicals then that we either purchased or synthesized. We could go back to bioassay. And what we could do is make use of an antibody against one of those effector proteins, AVR-PTO, and we got that from Greg Martin, another PRL alum. And what we saw is, we're not the first ones to think about, well, how does the bacteria see the plant? And what the field had been doing to this point and said, well, what's the apoplast? The apoplast is going to be acidic, it's going to be low nutrient, and there's going to be sugar around. And sure enough, if you do that, you do see a little bit of activation of the type 3 secretion system. But if you do that and add back 100 micromolar concentration of any one of these 5 organic acids, you see this massive burst of the type 3 secretion system. And pointing out the concentration, this is at or below what's known or what's been reported for the apoplastic concentration of all of these organic acids. And the key to all this is, if we take any one of these and we add it back to the MKP1 mutant, we completely eliminate resistance, we completely restore the ability of the bacteria to activate and inject its type 3 secretion system. And more importantly, if we would pre-treat the plant to induce PTI, these chemicals will completely suppress PTI just as well. So MKP1 genetically phenocopies a plant that's been pre-treated to activate PTI. And so from this data, what I feel confident is that we can argue that we've now identified what is resistance in PTI. It's rapid. That decrease in those metabolites occurs in about 1 to 2 hours. That's within the time frame of induced PTI. Completely stops bacterial virulence. The mechanism is bacterial static rather than cidal. And it's completely independent of SA, which has been one of the defining characteristics of what PTI is. The other thing is, it just makes inherent sense what's going on, because if you think about it, what the plant's doing is restricting nutrients. It's taking away the organic acids, the carbon and nitrogen from the amino acids that most invading microbes would be using. And it just so happens that the pathogens picked up on a few of these to actually activate its virulence program. So I just want to be clear that I'm not arguing that nothing else is happening, right? But I think this looks pretty clear to us that this is really the first chemical step that occurs in terms of stopping the bacterial growth. And then, as many things with the plant, it starts layering on other activities to try to maintain this resistance. So, of course, now the question, like everything in science, just kicks it down the road of, okay, well, so how does that happen? What controls the levels of those extracellular metabolites? Well, now that we had the borders, now we had some idea of what resistance was, we can go back to our pool of phosphoproteomic candidates. And one of the ones that really stuck out to us was this plasma membrane localized E3 ligase ATL6. Because what we believe is that what's changing is not the accumulation of all these metabolites, but the secretion of these. And we've done metabolomics looking at the entire plant. There's no difference in the overall levels of these. It's just not getting it to the apoplast. So this was attractive because we would think that perhaps this E3 ligase at the plasma membrane maybe is controlling the abundance of these transporters. So here's the pathology. If we infect with DC3000, the virulent pathogen, we don't see any difference in the bacterial growth, which would make sense. It's virulent pathogen. It's stopping the innate immunity, right? So we wouldn't see any difference. But if we pre-treat wild-type plants with flagellin to pre-induce PTI, we see this massive reduction in the bacterial growth, and we don't see disease symptoms. So as a control, we can treat FLS2, and not surprisingly, that when you don't have the receptor for flagellin, flagellin doesn't induce any of this. And that E3 ligase mutant is just as dead as the receptor. Right? We get absolutely no PTI being induced. So Occam's razor says, right, it must be affecting FLS2 levels. We go through protein levels for FLS2, normal, plasma membrane accumulation, normal, BIC1, the downstream phosphorylation events, normal, all the molecular responses are normal, transcript accumulation is normal. These mutants can do everything associated with PTI except PTI itself. So what we believe this indicates, in the easiest explanation, is that ATL6 must function fairly close to the actual mechanism of PTI. And so what we could do then is come back to our existing data. MKP1 is the furthest downstream genetic component known to be involved in PTI, regulating the regulation of the type 3 secretion system. So we made double mutants, and in seedlings, the double mutant with ATL6 completely suppressed the resistance in MKP1. That's also very true and very clean in adult plants as well. So completely epistatic, so it fits with the model that this is downstream. And then we developed a new assay where now we can infiltrate the bacteria into the leaves of the plant and then take leaf discs and look at the activation of that type 3 secretion system in planta during the infection. And what we see is what we had reported here using other methods previously, but in MKP1, the bacteria does not activate its type 3 secretion system, and ATL6 gets rid of that restriction and allows the bacteria to now be activated. Again, consistent with the pathology. So what we think now is that not only do we know what resistance is, but we think we have the complete signaling pathway. Because of the phosphorylation site here, we knew that this was not downstream of the MAP kinase 6 that's being regulated by MKP1. The sites were incorrect for MAP kinase-directed phosphorylation. So bioinformatically, we predicted candidates, and we've been able to confirm. I'm not showing it to you because it looks just like the ATL6. But this now brings in CBL, or calcineurin B-like protein, so calcium signaling is now integrated into this, and the CBL interacting protein kinases that are regulated by these look to be genetically, at least, the upstream kinases. So we think this is the complete pathway, and so the idea is, leading to the activation of E3 ligase, we either get mono or polyubiquitylation of the transporters for these organic acids, and getting rid of those gets rid of the type 3 inducing signals, and without those chemicals, the pathogen cannot become pathogenic. So in the wandering bit of science, like occurs, we were looking at this, and the big clear part that we don't have our hands on are the transporters. And part of that difficulty is transporters tend to be promiscuous. So we're not expecting there to be one transporter. We think that we're simply getting, but we're getting the regulation of that entire network. So having developed proteomics, we went back and did a proteomic screen to try to look for those transporters. And this has led us into a collaboration with David Mendoza at the University of Missouri, who works on iron, and Anteheza with the protein trafficking. I'll allude to also how she came into this as well. But the proteomics showed us that within 1 hour of PAMP treatment, about 3% of the enriched plasma membrane proteome was decreasing. So up to this point, what we had known was that the receptors themselves were rapidly endocytosed. It's part of Antea's work showing the desensitization that occurs during the infection. What we can see now is there's actually a really coordinated network of plasma membrane proteins that are being rapidly regulated. Now, a small portion of this list of these, PRL connections. Frederica, about a year or two ago, was at one of our meetings and saw our poster and pointed out, that's our protein. And so one ends up getting new collaborations and looking at that. We've also confirmed that these are rapidly decreased. But the one that really stuck out to us and jumped out is what we've already heard about, IRT1. And as we already heard, the reason that that was so striking is IRT1 is in the roots, and not only is it in the roots, it's only in the epidermal cells of the roots to interact with the soil. PRRs, these receptors for the PAMPs, are basically not present in the roots. It's only thought to be in the aerial tissue. So we're looking at this saying, well, how is this connected? And as Mary Lou had said, that levels of IRT1 are inversely regulated, that under high iron levels, it tends to decrease the levels of IRT1, prevent that excess uptake. Under low iron levels, it increases IRT1. And there's this tremendously complicated transcriptional network that occurs to control this. In this response for IRT1, and here's the confirmation that we can see within 1 hour, we're getting this massive reduction in IRT1 levels, and it continues to decrease. Here's another plasma membrane protein, just showing that not everything's there. Just sort of a shout out, if anybody works on plasma membrane proteins, particularly if you're doing proteomics, we developed this new method. It gets us around aqueous two-phase partitioning, which is ungodly awful, takes forever, hard to optimize. This method only takes about 3 hours, and so far we've used the same method on Arabidopsis mature plants, we've used it on maize, we've used it on maize roots. Basically, it works for everything and no optimization. It's a lot simpler to use. And what it's really doing is it's really depleting a lot of the ER when you end up going through, and that gives you then greater access when you're trying to do proteomics to sequence. But the important part here is during this response, there's no transcriptional change in any of the components involved in the regulation of IRT1. So this is a post-transcriptional activity. There's been a couple papers in the recent years where they were looking at transcriptional changes caused by PAMPs, but this is 24 hours later. This is coming in a completely post-transcriptional regulation that's occurring well preceding all of that. So as I said, we were kind of struck by this because, how is this all being put together? The receptors aren't supposed to be there. So we did a sort of a simple in concept, a pain in terms of dealing with small seedlings, but we could take Arabidopsis plants, and if we treat the intact plant with either water or ELF, and then bisect out the roots, we see that decrease in ELF. But if we bisect out the root and treat then with ELF, nothing happens. So it has to be perceived in the aerial tissue, and some unknown signal needs to be sent down to the roots then to cause the removal of IRT1 in this rapid type of response. So knowing that Mary Lou was going to be speaking is one of those cases where you go down weird paths on this. So went back and mined all of our omics data because we were thinking we should be looking for some small peptide perhaps in terms of something being sent. And actually our omics data came up with a good candidate, and it turned out to be very interesting for the wrong reasons. But what we found is what we think is this missing signal that controls the IRT1 levels under low iron conditions. So these are under low iron conditions. This causes accumulation of IRT1. We have a knockout of both the peptide candidate and its receptor, and IRT1 fails to accumulate under those low iron conditions. And none of the transcription factors are affected in these mutants. None of the IRT1 transcript level itself are not affected in this mutants. Everything happens properly. So we think we may have this missing long distance signal. We're doing now the complementation, the aerial tissue and the roots to see if we can dissect this more. But science takes you in weird paths that you end up working on things that you didn't think you were going to work on. And it all started with going to a lunchtime seminar by Doug Gage that takes you down. And it was really just sort of nice and amusing to see. I told Doug I was looking for a photo because I wanted to include something with him. And then you find out he's back here at Michigan State. And actually, I did not know that he was a postdoc at the PRL. All right, I wanted to come back and talk about some of the other things at the PRL. The science is important, but some of the other things that we picked up. Any of you that have done this, trying to navigate a career in science is tough enough. Trying to navigate a career in science with two of you trying to go through that career path, not necessarily straightforward. And as Antje and I were looking to do our postdoctoral work, we got some really good advice from Pam Green. And it's one of those things that sounds incredibly simple, but it's really profound. She told us you can only have one top priority, meaning that we were both driven. I mean, we came out of the PRL. We were both worried about our careers, but we couldn't be making choices based on what's best just for me or what's best just for Antje. We had to be thinking about how to make things work. And this has really stuck with us. We've been moving through Basel, then through England, now to the University of Missouri. And this just keeps coming up because it's a constant reevaluation about how do you make things work and what are your top priorities, and to have that discussion with each other to go through. And it struck us enough that we actually passed this along to our undergraduates, both in the classroom, but also the people that work with us. Because I do think in this career particularly, but it's true in many careers, that one has to decide what your priorities are. And so this has really been a guiding principle. Of course, the part of the PRL are all these close friends that we've had over all these years. Seeing Sharon's here, I didn't see you before with this, but we just had, not only was the science great here, but the people were just utterly fantastic. I point out Eric the Vampire Nielsen. I have so many photos of Christiana, but apparently your soul cannot be captured on film. So, that's not leaving you out for any reason, but it's just sort of shocking that, yeah, you have some weird properties. And we did great science, but we also had a lot of fun. I'm not sure if the tradition still continues, but we used to do the holiday skits where the postdocs would write a skit to make fun of the professors and the graduate students. So there's John Scott Craig, the old man, not only a longtime Dart friend, but also he had the unpleasant honor of mentoring a very green graduate student during my first rotation in John Walton's lab to try to teach me some molecular biology. So that was them doing the skit, and the really fun part was then I had a photo of Bob Mealy dressed as John Scott Craig, representing the ghost of PRL past, PRL present, and probably the PRL future, which tells you how long the old man was here in terms of the PRL history. We also had these skits. This was from a year, so this is Chris Rock, who was in Jan Zevart's lab, doing some rap, which I can't remember. But you can't really read it, but this was one of those years where Chris was being very inspirational, Chris Somerville. And so he was going on this rant about our goal was total knowledge, which we all found hysterical, so we had to take the piss in this whole thing. And so Antje and Sharon are being Pam and Natasha, the Fly Girls, for this rap in the background. And then also having some fun. Here's a photo of Christoph and myself. So again, from the 90s, if you watch Saturday Night Live, there was Hans and Franz, and the whole thing was, listen to me now and hear me later. We're going to pump you up. So we were then Hans and Jan pumping you up with plant hormones. So, most of the people, everybody here, has experienced this vagabond life that we have, traveling around, and the friendships that we build over this time are the most meaningful. Nikki was a graduate student in Pam's lab, and so wherever we had gone in the world, Nikki was kind of following us around in terms of visiting, going through. Antje and I finally got around to going on sabbatical last year, and so we were in Dusseldorf. I was in Wolf Romer's lab and Antje and Petra Bauer's lab. Michael Feldbrugge, who was also in Pam's lab, turns out to be the chair of microbiology at the HHU at Dusseldorf. And so we got to spend a ton of time with both him and Sigrun, who is also a postdoc at the PRL. And one of those dinners when we were over at their house, it was just this conversation of, how is this so easy? We don't see somebody for 20 years, but the bond and the trust and the friendship was here. It was just like going and having dinner with really old friends. Pauline, who is here, we used to have these cocktail parties. We were all dressed up and pretend we were grown-ups and going through. And when we went to go do our postdocs in Switzerland, Pauline was doing her postdoc actually in the French-speaking part of Switzerland down in Lausanne. And so we ended up doing a lot of different things together. We also had here always celebrated Antje and Pauline's birthdays together. And so we were in Switzerland, going in Europe. We did that again. We were just more fancy. So we got together and went hiking in the Swiss Alps, or we met in London for the birthdays. But here's when Liam was being born, and Pauline was a little freaked out because Liam had just kicked. And so Jenny was a postdoc in Ken's lab. And Tom, then partner, became her husband. And we've been close ever since. Jenny and Tom were the godparents for our son. And one of the things that we did at the PRL was on Friday nights after playing darts and pool at Coral Gables, we'd go back to Tom's place and play board games, European board games. And so when we were on sabbatical, Tom and Jenny came. So we could go. Essen is the oldest and the largest board game convention around. And so we'd always talked about doing this. And we went last year. And Tom was my best friend for 30 years because of this place. And he unfortunately, unexpectedly passed away this spring. And it's just a reminder of, this is something that I had from the back of science while I was a graduate student, one of those things you just happen to read. It was a comparison talking about the creativity in art and the creativity in science. And I took this and I had it taped above my desk. And I think it's just a reminder about perspective. I mean, science is deeply personal, right? We're committed to this. This is a big part of our life. But the perspective of this is that the work of an artist is permanent. Nobody's going to replace that, right? It speaks to the world. But inherently, in our role as scientists, whatever we discover is going to be supplanted. It's going to be replaced. And it's a reminder that I think that if we're in this business to try to create a lasting memory of ourselves, we're in the wrong business. And certainly the activities and this bit of reality crashing in on me this year really is a reminder that what I value the most out of the PRL are these friendships and the chosen family that we have as we go through this wonderful world of science. So with that, I will point out, since there is some science in here, the lab members involved. Martin has done all the work on ATL6 and the CBLs. Gabby's done the work on IRT1 and the proteomics discovery. Of course, everything, as I had just said, is built on things, and some of the preliminary data came from past members with our collaborators and funding from various agencies. And that's a recent photo of the lab, and we're in the age now of PRL, so we're in the age of AI, and so we can have the image of the world as they see us, and AI can show us then the world as which we think the world perhaps ends up seeing that. So with that, I'll be happy to take any questions. I'm really looking forward to catching up with many of you. So many dear old friends are here today. Happy to talk science, happy to reminisce, but it's wonderful to celebrate here with you on this great event.
Thank you, Scott. Wonderful talk. So I would like to open the floor for a couple of questions for Scott. Yes.
Fantastic talk. With your metabolomics analysis, two of the metabolites, the shikmate and
hydroxybenzoic acid.
seem to be related to SA, while SA seems to promote immunity. So do you have any perspectives why they seem to be the major players there for negative regulation?
So I would clarify, they're not the major players, right? The other ones have in terms of the other activity with that. That also told us that because they're from such very different metabolite pathways that we don't think it's any one thing being regulated. We did try SA independently, has no effect on the bacteria. So, coming back to this, I think my view of what evolutionarily happened was, and this is why I think it makes sense for PTI, I think this regulation was basically, I don't think plants in defense are as smart as some people say. If they see a fungus or a bacteria, it just freaks out and throws the kitchen sink at it. So I think all it's doing is retracting all these nutrients from the environment, figuring that's going to make it more difficult. And so for whatever reason, that some of these pathogens have now started to acquire this ability. That was part of my sabbatical. So Will Fromer's lab is now starting to look at some of these to see in other bacterial pathosystems whether these may also have effect. We think there may be an intersection with the sugar secretion that he's been studying. But I think it's just been sort of these things that acquire. The postdoc, Jeff Anderson, who did the work in my lab, is a associate professor out at Oregon State. And he did a really clever genetic screen, has actually found the bacterial two-component systems for a number of these, which really kind of connects the dots and says that these basically perception systems, I think, came later. The initial response was to remove these. At some point, these bacterial pathogens evolved the recognition in whatever way. And I probably wouldn't be surprised is if we started going to other pathosystems, there may be a non-overlapping subset of some of these signals. But we have some evidence that the paradigm holds up in other pathosystems.
How about non-pathogenic organisms, microbes? Any relationship between? Oh, sorry. Any relationship between these discoveries, you know, the small molecules, et cetera, and bacteria and fungi that either don't matter or do matter in a positive way?
So it's a great question because, of course, when you look at this, it looks like that's a great mutant, right? It was just like, so why does this gene exist in nature? So that was one of the hypotheses, and one of the other things I wanted to do in Wolf's lab was to look at the microbiome, right? Because one possibility is, so why isn't this permanently there? Maybe there's a negative effect on beneficial microbes. Like happens, I think we could take any mutant any of us work on, and you'd find some small alterations in a microbiome. I'm kind of a microbiome skeptic. But we didn't see any major changes for this. So at least in terms of these metabolites and that broader panel of nutrients were there, it didn't seem to have any large effect either on populations or in terms of overall numbers and or the distribution from the sequencing. So it was one of my favorite hypotheses. So what I actually think is one of the things that we got in with this, again, through friendships, so Roman Ulm, who's the chair down in Geneva, he was a graduate student when I was at the Friedrich Miescher Institute. These mutants also have, they're hypersensitive to genotoxic stress, so UV light. So I actually think the reason is probably there's some level of genome instability that would occur if the mutation existed, and I think that might be the selecting agent on it. But so far, we don't see any effect on the microbe, hand waving. But at some point, I have to try to go to sleep at night.
Great talk, Scott. So how about us who are working on crops that are being annihilated by pathogens that are introduced by insects directly into phloem? Any thoughts?
Biochemically, as you know, that's a pain in the butt to try to figure out how to study. I mean, Susie has done some really great, sorry, I should say Suzanne, but she's always Susie to me after the years in Hans's lab, trying to work on some of these things excreted and whatnot. We're getting into this now with single-cell sequencing to be able to start seeing where some of our candidates are, and we're doing single-cell complementation to start seeing where some of these pathways go. So I think all of those get a lot more complicated, and there's a reason. For instance, I chose bacteria and not fungi because fungi are very asynchronous. And biochemically, that just means that you're always looking at an averaged response. Whereas with bacteria, we can go bam and just give them a ton, and that's what we need to see the differential. But now that we have some tools, we can try to go in. That was one of our other hypotheses. There's been some anecdotal comments in the literature that resistance to biotrophic pathogens may come at the expense of against necrotrophic pathogens, so these things that would kill the plant. We've actually tried this with necrotrophic with fungi, and actually they're more resistant against necrotrophic fungi as well. Now, we don't know the full genetic range on that, but it's a great question, but that stuff is really complicated.